Okay, this is Animal Diversity, Chapter 1.5, Revisions of Darwinism and Evolutionary Theory. So, this first section, Neodarwinism, talks about some of the most real critiques of Darwin's argument, and that is that he never was able to identify a mechanism for inheritance. And so why is it that Lamarckian evolution isn't true? He didn't know what Gregor Mendel knew, although he did have some information from August Weismann, and basically he looked at animal development under a microscope, and he showed that he could separate cells that from the body, which we called soma, early in development, and there is no means by which the use and disuse of body parts could alter heredity. So, again, he's showing that Lamarckian evolution is bunk. It's something that you probably know today. Again, if you do a bunch of push-ups, your baby is not going to be any stronger than you, or than it would have been. So he showed experimentally that modifications of an organism during its lifetime do not change its heredity. But we know now, in part thanks to the monk Gregor Mendel, that organisms follow certain hereditary principle. And Mendel, who, by the way, didn't know about chromosomes, he didn't know about DNA in the way we know about it today, but Mendel laid it out for us mathematically that an organism's offspring is going to be a blend of the two parents. And there isn't any evidence for the fact or the notion that Darwin ever read any of Mendel's work. But if he had, it would have most likely been an aha moment from him. So in the 1930s, we had this new group of evolutionary biologists who were exploring evolution from a mathematical perspective, and they synthesized the ideas of genetics, population genetics, paleontology, biogeography, embryology, systematics, and animal behavior in what we call the new synthesis or Neodarwinism. So it says here on page 29, population geneticists study evolution as a change in the genetic components of population. With the establishment of population genetics, evolutionary biology became divided into two subfields. Microevolution pertains to evolutionary change in frequencies of variant forms of genes within populations, and macroevolution refers to the evolution on a grand scale, encompassing origins of new organismal structures and designs, evolutionary trends, adaptive radiations, phylogenetic relationships of species, and mass extinction. So, microevolution is a change in allele frequency among populations, and macroevolution is more speciation. It is larger. They're kind of two sides of the same coin. I've heard people argue they believe in one and not the other, but frankly, that's just kind of nonsense. Microevolution is change in allele frequencies. Macroevolution is larger speciation events, origins of new organismal structures and designs, evolutionary trends, etc. So we're going to move on to 1.6, which is a look into microevolution, and again, it talks about things called alleles, and it talks about blood typing. Okay, so if you don't know what your blood type is, you should find out. There are little kits you can get online. You just prick your finger and you put the blood on there, and you see which antibodies react. It's a really cool system. So we're focusing on this first row here. It says red blood cell type, group A, group B, group A, B, group O, and you'll notice group A has got these little circles on them. Group B have got these little rhombuses or squares or whatever. And then AB has both shapes. Group O has nothing on it. There are no antigens here, as we're called. And so why is it that A has got circles and B has got squares? Well, it has to do with the genetic control mechanism. So if you are a person who has the genotype for type A blood, you'll have circles. If you have type B blood, you'll have little squares. If you have an A and a B, you'll have both squares and circles. So what leads to this phenotype, your phenotype is your observable characteristics. Your genotype is what genes you have. So what we're looking at here is the phenotype. Polymorphism is the occurrence of different alleles of a gene in a population. And so MJC, we have polymorphism within this allele. Lots of people with A, lots of people with AB, lots of people with B, lots of people with O. All of the alleles of the genes possessed by members of a population collectively form what we call the gene pool. The allele frequency of a particular allele of a gene in a population is called its allele frequency. We're going to do some practice with allele frequency in another lecture. Because each individual genotype contains 2 copies of a gene, the total number of copies present in a population is 2n, or twice the number of individuals. So if it's me and 4 friends, if there are 5 of us, for this particular gene, there are, well, I'll ask you, how many total alleles are there for a gene amongst 5 people? Well, there are 10 amongst 5 people. Each of us got 1 allele from our mom, we got 1 allele from our dad, leaving us with a total of 10 alleles for one particular gene. We're going to talk about Hardy-Weinburg, I think, in a later video, but having different types of genes that code for proteins, hopefully you guys all understand that DNA is transcribed into RNA and then translated into protein. But having different genes allows us to have what is called protein polymorphism. Different allelic forms of genes encoding proteins that often differ slightly in their amino acid sequence. So you have different genes. Those different genes lead to different proteins. That leads us to what's called protein polymorphism. Now, we can actually study protein polymorphism. This is an example using this isn't a Western blot. This is just gel electrophoresis. But you can separate proteins based on their size. How large are the proteins? Well, you put them into something called a gel electrophoresis machine, and it will actually separate these things based on what size they are. And if you want to further study them, you can use a technique called Western blot to check for the presence or absence of proteins within a sample. So if you're doing forensics, you find some blood on the floor and you want to know, well, what kind of proteins does this blood possess? You can do a Western blot on it, which does start with gel electrophoresis. We also have a notion of genetic equilibrium, and we're going to use something called the Hardy-Weinburg equilibrium. It's an equation that we're going to use to study it, and it allows us to estimate the relationship of frequencies of alleles at a genetic locus and the frequencies of genotypes formed by random fertilization of gametes with respect to this variation. You can really only do one locus. You can only do one set of genes at a time. So, for instance, you could do blood type. But Hardy-Weinburg allows us to draw some pretty powerful and interesting conclusions. And I think in a separate video I'm going to go ahead and do the practice problem for Hardy-Weinburg. I guess in that video or in another video I will also talk about the characteristics that you need to have happen within a population for Hardy-Weinburg, as well as the forces of evolution. Stay tuned.